what we want to focus on today. We talked about natural selection. Natural selection starts with what? Populations have what in them? Variation, right? And in variation, some traits are favored by the environment compared to others. Those traits are selected for, while other traits are selected against. Therefore, the alleles, if I'm talking about Darwin's finches, right? If the ancestral finch lands in an island with a larger food source, and that food source is larger, the birds with the larger beaks, slightly larger beaks, are going to be favored. They're more likely to survive and reproduce. Therefore, those alleles will go, that allele frequency will go up in the population, and the birds with the smaller beaks will tend to die off. That allele frequency should go down in the population. Okay, that's where we need to be right now. Is everyone okay with that? So we're doing natural selection. That's all natural selection is. It's really a fairly straightforward concept. What we're going to talk about today is a little bit more into that gene level, the allele level. We're going to talk a little bit about how natural selection can affect populations, looking at graphing natural selection. And then we'll talk a little bit about Hardy-Weinberg equilibrium. There's our friend Darwin on the left. We haven't done this yet, right? Okay. There's our friend Darwin on the left. And on the right is Gregor Mendel, who very much looks like a Gregor. Gregor Mendel. Darwin had no idea what Gregor Mendel was doing. Gregor Mendel had no idea what Darwin was doing, right? Our modern view of natural selection, which is known as modern synthesis, is really just a combination of both of their concepts. So modern synthesis is just a combination of both of their concepts. So basically, everything we're talking about today is really modern synthesis, applying natural selection to our understanding of genetics. Okay. We're going to spend a little time on this slide. This is really just to kind of understand some definitions. That's the whole key, but we're also playing, little calculations here. Gene pools is the combined genetic information of all the members of a particular population. Gene pool is defined by population. A population is the same species living in a given area. Before we go any further, we need to figure out what do we mean by species? How do we define species? Seems like an easy question, right? At this point today, it seems like, well, we should be able to define a species. Not quite so easy. The definition we're going to go by is the ability to produce viable offspring. So species would be two organisms of the same species would be able to produce a viable offspring. Now let's talk about two organisms that are not the same species to highlight this. A horse and a donkey. I cross a horse with a donkey. They can reproduce. They produce an organism known as a mule. Can mules reproduce? Mules are infertile. Horses and donkeys are not the same species because a mule is not a viable offspring. Doesn't mean mules don't exist. Right? That's not what viable. Mules exist. Right? It's not like a unicorn. But they're not viable because the mule species themselves cannot carry on. They cannot go on any further. The only way we get mules is if we cross horses and donkeys. If we got rid of all the horses and donkeys and had a whole bunch of mules, the mules would last one generation, and then they would die out. Right? Is this going to progress us forward?
What's the point of mules?
Mules are a hybrid, so they have a little bit, donkeys tend to be a little bit stronger and hardier in terms of carrying. Horses are a little bit faster, I believe. So mules kind of that good hybrid between the two. So it's a hybrid of traits, something to that effect. Okay, so that's our first thing we need to get in this slide. Definition of species are organisms that can produce viable offspring. Now that seems like a pretty easy definition. How do you define two different plant species though? Because plants are a little strange because sometimes plants of different species can actually reproduce and produce hybrids. So you can see how that definition falls apart when we start talking about plants. And then we talk about bacteria, they reproduce asexually. So therefore, how do we define them as a species? So the species concept is actually harder to determine than it seems. We can do it through DNA testing. Like we can do like DNA similarity, but us and chimpanzees are like 99% the same. So at what point do you have enough change in DNA to say that you have a different species? Think about bacteria. Like, trying to classify bacterial species. Like, how do we know these bacteria are the same species or different species? Is it just variation within the species? Or is it similarities between two different? It gets really, really complicated really far. Hence, when we classify organisms, we don't classify bacteria that level. It's hard to do that. At least not down to species level. All right, so that's kind of our species talk. The next part is we said gene pool. A gene pool is when you have a population that is breeding together. If I have the squirrel population in Central Park, and the squirrel population, what's the park in Fort Lee? Fort Lee Park? Byron? Byron. Okay, so the park in Fort Lee, whatever it's called, and Central Park, they have squirrels. Do you think those squirrel populations are interbreeding with one another? No, so they would not be the same gene pool. They would be separate gene pools. They could be the same species. They might be able to produce squirrels, but they're not the same population because the Hudson River is preventing them from breeding with one another. Okay? So gene pool implies that there's a group of organisms that's a population. A population means living in a given area and they're breeding with one another. All right, next definition. Relative frequency. We've already kind of talked about this, right? Relative frequency is just the frequency of the dominant or recessive allele, the frequency of any allele in a population. That's all it is. So relative frequency is just a frequency of an allele in a population. That's it. We are going to talk mainly about dominant recessive situations. So if the frequency of the dominant allele is 60%, the frequency of the recessive allele must be 40%, right? It's all percentage. So all should add up for 1. Everybody good? So here, I'm looking at a mouse population. Dominant recessive trait. The black trait is dominant to brown. And we have 48% of the population are heterozygous black. 16 are homozygous black, 16%. 36% are homozygous brown. We can actually look at those and actually calculate allele frequencies. We'll be able to do that later. But for right now, we have this nice little chart at the bottom. Each dot represents an allele. What's the frequency of the brown allele? Count it up. Count on your computer.
How many brown we have?
What's the frequency of the brown allele?
29.
It should be 30.
Yeah, 30.
That means the brown allele makes up what percentage of the population? There are 50 there. 30 or 50 would be 60%. So the brown allele makes up 60%. Now we can count the black allele, but we can just really go 40%. So the brown allele is 60% of the population. The black allele is 40% of the population. Everybody okay with allele frequencies?
Yeah.
All right, that's it. Easy. How many mice are represented by this population right here?
25. What, right? What? Right?
How many mice are represented by those alleles, by that population right there?
Any of 50? 25? Is it like 7?
It's 25. Why 25?
Oh, it says that you need to.
Every organism gets 2 alleles for every trait. So if there are 50 alleles there, it represents 25 mice. How many alleles do you have for every trait? 2. So therefore, if there are 50 alleles there, each mice has 2 of them. So it means it's 25 mice represented there. Everybody okay? All right. We're good so far. We're just getting some baseline stuff. This is just a slight sidebar, but just kind of like making the point again. What's the ultimate source of all variation in populations? Mutations. Now, what helps to increase variation as well is gene shuffling, right? Meiosis and crossing over help to increase variation. But the ultimate source of variation are mutations. Everybody okay with that? I'm going to use the good old card example to highlight why. Gene shuffling is like taking a deck of cards and shuffling it up a little bit. That's what gene shuffling and crossing over does. When you cross over, do you get new alleles? No, you just kind of mix up existing alleles. That's like shuffling a deck of cards. When you get a mutation, it'd be like adding a different card to the deck, like a new card, a new number. Everybody understand that? So gene shuffling helps to increase variation, right? Crossing over, part of gene shuffling helps to increase variation. But the ultimate source of new variations, of new variants, are mutations. Think about it. When we did the fruit flies and parental types, recombinant types, did the recombinant types have different genes in the parents, or were they just mixes of the parents? They were just mixes, right? They were recombinants, a mix of the two parental types. Okay. Everybody okay? We are moving. We are rocking and rolling today. I feel good. Yes. Yeah. What? So the card example is this. So if meiosis and crossing over is like shuffling the deck of cards. There's no new cards in the deck. We're just getting different combinations. A mutation would be like making up a whole new card. So we're going to make up card 6.5, and we're going to make it triangles, and we're going to make it purple. Exactly. Exactly. So the ultimate source of variation is mutations. How new genes get introduced to mutations. Okay. Many traits that we look at are single gene traits. Single gene traits are pretty easy because you really just have two phenotypes. Like if you look at our mice, they were heterozygous, homozygous, dominant homozygous recessive, but when it comes down to it, they were either brown or black. So when we look at single gene traits, the best way to kind of graph it would be a bar graph. Right? The mice would either be brown or black. If the brown mice frequency went up, the black mice frequency would go down. Widow's Peak is another one. Anybody have a Widow's Peak? I go like this all the time. It's so funny. There's nothing to hold back. But Widow's Peak right here. Anybody have a Widow's Peak?
Do you have a widow's peak?
I do. Actually,
I think I have one.
It's
It's always this big.
So a Widow's Peak is it. Don't worry, we'll play with like the Widow's Peak thing later. But it's dominant recessive, so it's pretty easy to grasp. However, most traits are more complex than this, so therefore we can't really use a bar graph. We use a bell curve, such as human height. Could we group human height into 5 categories? Not very accurately. How about 10, 20, 100, 1,000? No, because it's all longer continuum is what it is. So human height is a longer continuum. You can really be anywhere within a range. So how we would graph it would be more as a bell curve, which the center line would be, 5'5" to 5'7" would be kind of most people would fall in that range. On the low end, down to like 3'2", not too many people 3'2", 7'6", not too many people 7'6", but it'd be more like a bell curve. Everybody okay? What's that? That's what we're going to get to. Yeah. So we're all okay so far? Okay. What leads to variation? We just answered this. Mutations and gene shuffling. How does the range of phenotypes differ between single gene traits and polygenic traits? Single gene traits are really the word you want to use. It is dimorphic, meaning 2 possibilities, dimorphs. So I won't use one dimorphic, but dimorphic. And with a polygenic trait, it would be dimorphic, it would be polymorphic, right? Many morphs, many types. Okay. I'll be quiet for a second so people who are taking notes, I'll let you copy this down. So you can get the bulk of your notes here for the next 5 or 10 minutes. But no, I'm going to give you a couple minutes. For the next 5 or 10 minutes when I talk, you won't have to write. Yep. Yes, just say ID and planner. What's up?
So recently my phone hasn't been working on battery. It was like my phone was shut off. So I went to Apple and it turned out I had a swollen battery and it was pressing up against my screen. Right? And so I was like, oh, so you're just gonna switch my battery, right? And they're like, no, we're gonna give you a new phone. I was like, why? And they said swollen batteries are explosive. So I was technically carrying around an explosive phone.
Explosive device?
Yeah.
That's crazy. You got a new phone?
Yeah.
Did they give you a newer version?
No, it's
Okay.
they never did that. Yeah,
That's game.
But I was bringing like an iPhone 1.
Yeah, the version at least she didn't go on the news saying her phone is low.
Just kidding. They'll probably just like
All right, everybody, just take a second, copy this down.
You guys are supposed to be furniture thing.
never gonna be because of cakes. Breaks gonna give you
Kevin don't. Oh, it's so easy. Definitely. Wide radius. Division.
I was hoping you would Strangers, have you ever been that stressed?
Why is this stress?
You're just sitting there for like this.
What's going on?
No, no, he just he's stressed out because it's all his
What's up?
Uh,
For a minute. Actually, I have a meeting, so it has to be like, give me 10 minutes, and then I'll be back.
It's short. I probably should. While you were mentioning with the kids.
We might have time. Okay, everybody got the notes down?
no.
All right, so let's talk about this. This is how natural selection acts upon population. These are 3 ways in which natural selection can act upon a population. And again, we're focused on here polygenic traits. Directional, stabilizing, and disruptive. We're going to use the finches as an example. It really could be anything though. Shifts in color, shifts in size, anything like that. What we have is we have an ancestral finch species that lands on a new island. New island has a food source of larger berries, right? So what happens is the birds with the larger beaks, the yellow, are more fit in that environment, meaning they are eating better, they're more likely to survive since they have a low mortality, and since they're going to survive, they're more likely to reproduce since they have a high fitness. Yet they have a high fitness. So what happens over time is the allele frequency for the large beak alleles, those allele frequencies are going to go up. At the same time, the birds with the smaller beaks have a higher mortality, less likely to eat, less likely to reproduce. Those alleles dictating smaller beaks will go down. Over time, you can see what happens to the average beak size. It gets larger. Notice, though, it still maintains a bell curve shape. Now, in nature, is it always a perfect bell curve? No, but it does represent somewhat of a bell curve, yes. Fitness is the ability to survive and reproduce. So here's our initial, and then the dotted line is representing this right here. And then over time, you can see the beak size shifting towards a larger size. Could it shift towards a smaller size? Yeah, we just change the environment. Directional selection could shift to the left here, towards a smaller beak size as well. It could be for height. It could be for color. It could be for a number of different traits. Question?
So, like, would it start as a side, like, on the left, would be, like, it would go from one to something.
What do you mean? What do you mean?
Like, if you said that the beak size increased, would it always go into
Well, I mean, so put it this way. Beak size would increase until you get into an equilibrium state where the environment no longer selects for the larger beak size. And at that point, it would kind of stabilize and stop shifting. We're just going to hit an equilibrium state. If you change the food source and made a smaller food source, it might start shifting back the other way. So it depends on the environmental pressures. Yeah. There's two examples for stabilizing. Fitness lies in the middle, means the average is the most fit. The extremes are less fit. The number of eggs birds lay in a nest is a great example of this. Birds could lay 20, 30 eggs if they wanted to, right? Like sea turtles. Sea turtles do that. The sea turtles take care of their young. Do you see sea turtles coming back to take care of? No, they're on their own. They hatch. They try to make it to the water. Half of them die, if not more, right? And it's kind of
That's like laughing.
if they die, they die. Birds, however, have parental care, right? So when the birds hatch, if there's 20, 30 birds in the nest, the mother or the father bird have to bring back enough resources, food, to feed 20 or 30 birds. That can be difficult to do. Therefore, the entire, all of their offspring might be weakened, and they might be less fit. In the reverse, they might only have 2 or 3 eggs. Well, birds are preyed upon. Having only 2 or 3 eggs makes a real possibility of all of them dying off. So what is most fit for birds? Having somewhere in the middle. Right? Not too many eggs, not too few eggs, but kind of a stabilizing selection, having an average number of eggs. So around like 6 to 12 eggs might be a good number. Now all birds are different. Not all birds have 6 to 12 eggs, depends on what they do. But 6 to 12 eggs is a good number to go by. Right? It's stabilized. The extremes have been selected against. The average is now selected for. So it means overall they tend to have an average size number of eggs. The other thing with this is think of early human history, right? Not with our medical advances these days in hospitals, but birth weight in babies. There used to be a greater range in birth weight in babies, from 2 to 3 pounds up to 15, 16, 17 pounds. Before hospitals, that was a major issue for C-sections, things like that. So babies that were really, really tiny were selected against because there's a good chance they weren't going to make it. They weren't going to be strong enough, hardy enough to survive the elements. Babies that were too large, there were complications during childbirth that would cause major complications there. So it was selected against large and small babies. So babies have stabilized that average birth weight of like 5 to 10 pounds. Yeah, you go a little smaller, a little bigger, but that's kind of like the standard range. It's stabilized over time. Right? Here you see the curve squeezing the middle. So the bell curve maintains, but it just maintains the extremes are dropped off. So it squeezes the middle. The last one's disruptive selection. My finches, going back to our finches, that land on a new island that has multiple food sources. Some of the finches migrate up into the canopy and eat the berries out of the tree, so they're favored to have larger beaks. Other finches stay on the ground and feed off small insects and maybe some leaf cuttings or something like that. So they're favored by having smaller cutting beaks. So now we have a separation in the population. Some are trending towards larger beaks, some are trending towards smaller beaks, because both are fit. So here, fitness lies on both extremes. What's not fit is having the average size. The average size, you're not good at either one. So the average they tend to die off, and the allele frequencies for the large and small sizes will increase. You can see we get a separation in our bell curve. This is where we get a break, and we move away from the bell curve. And this eventually could lead to what? This could possibly be a speciation event, the formation of new species, if they separate themselves. There's a lot of things that happen, but it could happen.
You're like visually seeing natural selection. That's so cool. Because it's like the bell curve and it's just like splitting.
Like parting of the seeds. Question? Yes. It could. Everybody okay? Question?
Like if
I'll slow down. I'll pause here for a second.
if the environment changes, like the middle becomes more fit down,
They come right back together.
like they become one thing. They won't like stay the way they
Listen, the thing about natural selection, it's not like there's this progress towards this end goal. Natural selection is dictated by the environment. The environment's changing all the time. So the game is changed on a daily, almost minute by minute basis. It's always changed. Yeah. Just give everybody a second to catch up here. Say that again? What do you mean by that?
Because like the average for the disruptive selection or the extreme for disruptive selection, they become more fit, right?
Right, so you're saying more fit.
Yeah.
Yeah.
But for directional selection, it's moving in what direction towards.
So it's so one extreme is favored. In disruptive selection, two extremes are favored. Don't worry about it, we're not doing any pedigrees or anything.
No.
Okay. Are we okay so far? That is how natural selection affects populations. The next question is, Abdullah,
Yes.
does natural selection occur all the time? What?
No.
No, it does not. So now what we need to do is shift away from how natural selection affects populations. Like, well, what happens when natural selection does not occur? Or even better, what does it look like? What's happening when natural selection is not occurring? Good question. This is what has to happen. So again, I'll give you, this is a moment to take down kind of, this is what I'm going to talk about for a few minutes here. I'll give you a moment to catch up. Don't worry, we'll do something fun in a few minutes. Just hang with me.
What do you mean? This is so much fun.
What's that?
All of this has to happen. I'm going to talk about it. Yeah, you have to try. Is everybody okay?
Yeah, I want to put it down. Yeah.
Everybody's okay? We good?
Okay.
What we mean by genetic equilibrium is we do not have changes in allele frequencies. All of these situations here, all of these situations are implying changing allele frequencies. Changing allele frequencies. Changing allele frequencies, okay? Well, we want to focus on a situation where allele frequencies are changing, they're static, they're maintaining a state of equilibrium. Well, in order for that to occur, we need to meet certain criteria. One, there needs to be random mating. There cannot be a selection process. It doesn't happen with many higher level organisms, right? If you look at many mammals, there's a hierarchy in their society. You look at birds, birds are selecting on song, on color, right? So this is really kind of more for lower level organisms. Not that it can't be in larger organisms, because you can kind of look at individual genes. But in any case, that's what it means by random mating. It means there's no selection process. Think of a plant. Plants don't choose their mates. They just put pollen in the air. That's about it, right? So random mating. Populations must be very large. That's because of genetic drift. I'll talk more about this in a second. That's what the next slide is. Large populations, let me give you the simple one first. If I flip a coin twice and get 2 heads, does that tell me I'm going to get 100% heads from there on out? No, but it was a small sample size, so I could skew away from what probability tells us. Now, if I take it, I have 1,000 coin flips, Am I going to be most likely closer to the 50-50 breakdown, 500-500? Yeah, the larger the sample size, the closer you'll be to what probability tells us. So in order for our allele frequencies to maintain a static state, you can't have a small population because random changes can fluctuate. You need to have a very large population to kind of maintain our allele frequency. Okay? Okay. There can be no movement in a random population. If I have a population that's in genetic equilibrium and all of a sudden 5,000 birds now enter the population, do you think the allele frequencies are going to stay the same? No. Or 5,000 birds leave the population? No. So you can't have major movements in or out of the population. No mutations. Really, no mutations goes with natural selection. What do we mean by that? Can we stop mutations? No, they're going to happen. But a mutation can't make one portion of the population more fit than another portion of the population. So really, mutations is talking about natural selection. Obviously, if we're having natural selection, are allele frequencies staying the same? No, we're moving in some direction. If we meet all of these criteria, we can say that we are in a state of genetic equilibrium. A little bit more on genetic drifts. So the first type of genetic drift is what I just said. In small populations, you can skew away from what probability tells us because of randomness, random events. Right? Could, how many people here have brothers or sisters? How many people have only brothers? Right? Does that mean if you have another brother or sister, are you going to have another brother? Are you going to have a sister? No, it's randomness, right? Now, if your parents were to have 100 kids, most likely they're not going to have 80 boys and 20 girls. They're going to be close to the 50-50, right? That's what we're talking about. The other examples of genetic drift, these are also examples of genetic drift, but they're a little different. The first one is the bottleneck effect. It is exactly as it sounds.
Started out so low.
Skittles. Red, yellow, purple, green, orange. That's it, right? Okay. I take a 2-liter bottle. I empty it out of Sprite or whatever it was, and I put in Skittles.
Don't make
Don't make fun of me.
they flow. Like how much cooler can they get?
Art is in the eye of the beholder. I see art. I don't know. All right, so Skittles. Let's pretend when you get a bag of Skittles, there's an equal and even distribution. So let's say it's the same number of red, yellow, purple, green, and orange. It's not the case, but we're going to pretend that. All right, I put them in a 2-liter bottle. I tip it upside down real quick. 10 Skittles come out. That's it, compared to the hundreds that are in there. When I dump it out, I happen to get a whole bunch of reds, a couple yellows, a purple, and a green. Do the allele frequencies after our bottleneck effects, hence bottleneck, not all come out, get it? Bottleneck effect, do the allele frequencies match up to our initial allele frequencies? No. Did our variation increase or decrease? It decreased. That's what happens with the bottleneck effect. You have an initial larger population with a lot of variation. Some event kills off a large percentage of the population, leaving a smaller population with less variation. Yes? That's perfect. Yeah. So natural disasters, such as drought, or earthquakes, or tsunamis, human disturbances such as overhunting or population growth, right? As we start increasing and taking over habitats, right? Habitat loss. The organisms I like to talk about this are the cheetahs. How do you spell cheetahs? E-E? What?
Yeah.
That's right? Cheetahs. There used to be a lot of variation in the population, but because of drought, because of hunting, because of habitat loss, because we're building up and building roads that separates population, a lot of the cheetahs died off. And when a lot of the cheetahs died off, there was a bottleneck effect. They all died off, right? And only a few cheetahs were left. And the few cheetahs that were left had far less genetic variation. This is why the cheetahs are in danger, because they are so genetically similar to one another that any type of disease could wipe out the entire population. There's a lot of bad recessive traits that are showing up in the cheetah population because of this. So the cheetahs have experienced a bottleneck effect. Everyone get a bottleneck effect? Example, was that natural selection? No, it's kind of like a genetic drift. It probably falls in that category. The last one is a founder effect. Here's your founder effect. Here's my initial population of beetles. Lots of variation, brown and yellow and orange and reds. Now, the population, something happened, the population decided to leave. These 4 beetles didn't get the message. They didn't get the memo. So they stayed behind and started their new population. And once they left, population B said, hey, you guys are jerks for doing that. So they left the population and started their own population. Now, we have new populations, which are founding populations, hence the founder effect. And the allele frequencies in the founder populations, do they match the initial population? No, they do not. So we need to account for this. When we call it genetic drift, we call this the founder effect. The change in allele frequencies wasn't due to natural selection. It was due to a founder effect genetic drift process. Everybody okay?
Okay, so when you say for genetic variation, 5 conditions that lead to that, there can be no movement into or out of the population that's genetic drift.
It's kind of a genetic drift aspect. Yes, you can kind of group those together. Okay, now why do we talk about this? Why do we care about this? Well, because these 2 guys named Hardy and Weinberg, and they came up with the Hardy-Weinberg principle. What the Hardy-Weinberg principle is they said if a population is in genetic equilibrium, if all those criteria were met, no genetic drift or anything, there should be no change in allele frequencies. And that state would be a state of genetic equilibrium, or Hardy-Weinberg equilibrium, you sometimes refer to it as. Here's what we're going to do next. I'm going to go back. That's what we're going to work with. We're going to do some math. Okay. As people are finishing taking their notes, what I need you to do is this. I need you to tell me whether your earlobes are attached or unattached. What I want to know.
Yours look attached or attached. They're attached. Okay. Just anything we need to copy down. Was it like that one before? Oh no.
I think that's a complete
Get it.
it's like an outline.
Okay. Now, as you're assessing dominant, or you're assessing your earlobes, I need somebody to look up. What's dominant? Is attached or unattached dominant? Somebody Google it.
Attached is dominant.
Attached is non-dominant. I mean, recessive.
Michelle.
Yeah.
So who Googled it? What's dominant? Attached or unattached? Detached is dominant?
Yeah. Pretty sure.
No, I need to know for sure.
Oh.
Yes.
Yeah, dominant is unattached. Yeah.
Detached, unattached is dominant. Okay. All right. So now what I need you to do is, by talking with each other, I need you to tell me what is the frequency in this classroom, in our population, what percentage of us are homozygous dominant, what percentage of us are recessive, homozygous recessive, what percentage of us are heterozygous. I want to know the frequency of the dominant allele in this class and the frequency of the recessive allele in this class. That seems pretty ridiculous from very, very basic information, right? Like, how can we possibly do that by just looking at our ears? Guess what? You don't need to know anything about your parents. We could do it with this. This is the Hardy-Weinberg equation. If you stick with me, don't get frazzled right now because it's not that hard. It might seem hard at the beginning, but it's actually really easy. You will be fine with this. The Hardy-Weinberg equation is based off a heterozygous cross. Heterozygous cross is simply this, a dominant recessive cross, like we do with our pea plants. Right? We do our cross here. Big T, big T, big T, little t, big T, little t, little t, little t. Math geniuses. What's another way to write big T, big T? What's another way to write little t, little t? All right, we can't do anything with these. But we don't want to use random letters every time we do this equation, so we need to standardize it. We standardize it with P for the dominant trait and Q for the recessive trait. So P squared stands for homozygous dominant. Q squared stands for homozygous recessive. P and Q stand for heterozygotes, but when I do my cross, I don't get one of them, I get two of them. So this is the frequency of the homozygous dominant genotype. This is the frequency of the heterozygous genotype. This is the frequency of the homozygous recessive. Okay. Now, when you look at your ears are either attached or unattached. So we need to look at your earlobe. Your earlobe, I think I'm attached, right?
Yeah.
I'm attached. So attached, your earlobe kind of comes down, and there's not much of a gap between the bottom of your earlobe and your head. Unattached, it kind of really loops back up. You almost have like black skin waving down there. So what we need to do, are you unattached? Yeah, unattached. So stand up and show everybody. Pull your ear out. So unattached. So what I really need to do, I need to get an accurate count here. I need to know how many people are attached and unattached. So figure it out at your table. Figure out what you are. In about a minute, I'm going to have people raise your hands if you're attached, raise your hands if you're unattached. So know what you are. Heterozygous is heterozygous. So you would show you unattached. You have to have somebody show you. Yeah. Well, you have to know. What are you? Okay. All right, let's go. Take the ID.
Yeah, a lot of your
30 more seconds. I just told you Q squared stands for the frequency of the homozygous recessive gene. Homozygous dominant phenotype. No, heterozygous. Heterozygous, it shows the dominant phenotype. Yeah. All right, here we go. Raise your hand. Raise your hand if you have attached earlobes. Raise your hands. Nice and high. 8 plus Devin is 9. Raise your hands if you have detached.
Oh my god. So
Plus 7.
All right, good. Okay. Now, let's see how smart you are.
Why not?
Okay. Both the number 9 and the number 14, both of those numbers are important. We're going to use them to begin with. But which one is more important? Which one gives us more information?
14.
9.
14.
9,
I
maybe.
think it's 9.
14.
Okay.
Usually just
Why do you say 9 or why do you say 14?
But I don't know.
Be-
It's the 9 because 9, I mean, like, you know for a fact that they're both, that everyone, that the 9 people are always recessive, like, they're both, they're homosexuals.
I can't.
9 is most important. Why? Because 9 is the recessive trait. So these 9 people, you automatically know that you are homozygous. So we know your genotype. Do we know the genotype for the other 14? No. All right. Now, we already know what Q squared equals. Because Q squared is the frequency of the homozygous recessive genotype. What is the frequency? How do we calculate it from our numbers?
81.
Oh, that makes sense, yeah. Oh, yeah. Yeah. I was like,
We need your calculators.
my phone is recording.
Oh.
And the total of them.
Julia?
0.39.